Hey guys, welcome to the lectures that I was the most excited over the course of this semester to make. And those are going to be starting the topic of metabolism and glycolysis, Krebs cycle, pentose phosphate pathway, electron transport chain, all of that cool stuff. It was my absolute favorite thing. And I really hope that some of these videos will help clear up some of the intimidation factor, I guess, that goes into thinking about these things, because I know it seems very, very scary to look at a blank glycolysis diagram, or especially a blank pentose phosphate pathway or Krebs cycle, and think oh shit, I have to know all of those things. But it's really it's really not that bad. And it all works together and flows together very, very intuitively. And so if you draw out glycolysis a couple times, and if you can understand why the enzymes work as they do, and why certain structures are named certain things, you'll have a really, really easy time moving forward in this class and in any other class that is contingent on your knowledge of these things. And so what I wanted to do first was just go through, the steps of glycolysis. And so essentially the point of glycolysis is we're starting with a glucose 6-phosphate, or I'm sorry, we're starting with a glucose, and we're eventually breaking it down to 2 3-carbon compounds, two pyruvates. And so this right here, we've got glucose. And our first enzyme, and the reason that this enzyme does this is, we want to trap it in the cell. So we use hexokinase, and if you remember, the term kinase means that we're generally going to be dealing with a phosphoryl group, or we're transferring, a phosphate to something. So we're going to use ATP. So the enzyme is hexokinase. So hexo meaning it's a 6-carbon sugar, kinase meaning it's going to attach a phosphate. So hexokinase takes a phosphoryl group from ATP. We're going to make ADP, and we're going to release a proton. That proton is actually very important, so just keep that in mind while we're doing this. And so we get glucose 6-phosphate as our product of that reaction. And so you can see on the 6th carbon, we've got a phosphate group that kinase stuck on there. Next enzyme, phosphoglucose isomerase. And so this one also does exactly what it sounds like it would do. It's going to take a glucose that has a phosphate on it, so phosphoglucose, and it's going to turn it into an isomer of itself. So fructose is, another isomer of glucose. It's got the same molecular formula, but it's rearranged in a certain way. So we're taking glucose 6-phosphate and we're turning it into fructose 6-phosphate. And our next enzyme is actually this is the major regulatory enzyme of glycolysis. This is the one that, is usually associated with regulating this whole process. So this is a really important one. This one is phosphofructokinase. And exactly like the name would have you believe, it's a fructose with a phosphate on it, and it's a kinase. So we're going to add a phosphoryl group to it. So again, we're going to take ATP, and we're going to release ADP, and an H+, so that we can put this phosphate on that carbon 1. And so this is called fructose 1,6-bisphosphate. And this is You've probably seen this a couple times before, especially when you talked about like phosphatidyl inositol 4,5-bisphosphate and things like that. But this bisphosphate, the bis instead of like a di or another term like that, that has to do with the fact that these phosphates, there's two phosphates, and so they're on different carbons. If they were on the same carbon, then you would have like how ADP is called adenosine diphosphate. If they were on the same carbon, you would call it fructose 1,6 diphosphate. But since they're on different carbons, we call it bisphosphate. That's just where that term comes from. And so our next enzyme, so we know that we're going to be splitting this up into two different things. And so generally we have, there's no like real way to think about this. I don't think. I just, you have to remember that the enzyme is called aldolase. And so after we get aldolase, we split it into a glyceraldehyde 3-phosphate. And we get a dihydroxyacetone phosphate. And I'm not going to have enough room to write it here, but this enzyme that does this rearrangement, that's called triose phosphate isomerase. And it's also called, you'll see this abbreviated as TIM. I don't know why they call it that, but that's just how they've decided to abbreviate that. So if you see TIM, they're talking about triose phosphate isomerase. And that's named, triose phosphate isomerase because it's dealing with two sugars, triose, 2 3-carbon sugars. And they both have this phosphate on them somewhere, and it's just, a rearrangement of those two sugars. And so essentially what happens here is that we want both 3-carbon sugars that we proceed with to be in the glyceraldehyde 3-phosphate form. So we're going to take 2 of these, and we're going to continue the rest of the cycle with 2 glyceraldehyde 3-phosphates in mind. So now what we're going to do is this enzyme right here is called glyceraldehyde 3-phosphate dehydrogenase. And so the dehydrogenase part will tell you that this is a redox reaction. And so if it is a redox reaction, then you know that you need some kind of other, in this case it's a oxidizing agent, because we're oxidizing our glyceraldehyde. So what's going to happen here is we take 2 NAD plus and 2 inorganic phosphates, and we create 2 NADHs and 2 H+. These NADHs are actually really important, so keep those in mind. For not this cycle, but you'll care about them in the electron transport chain. And so essentially what happens here is we, instead of like having a kinase come in and do this, and possibly spending another ATP, we put another phosphate on glyceraldehyde without, expending any extra energy. We just used an inorganic phosphate group. So this is called 1,3 bisphosphoglycerate. And again, bis because they're on different carbons. So we've got 2 of these. And so our next enzyme, now this one is called phosphoglycerate kinase. So you know that there has to be some sort of phosphoryl movement going on. And specifically that movement is going to be this phosphate coming off of it. So we're actually going to be taking two ADP and we're making two ATPs. And this is a really important reaction to have happen. And there's like actually an action center question. There's two action center questions that focus on this. This reaction is really, really important because without this reaction, we can't pay back the two ATPs that we invested into running glycolysis. So our next structure, this one right here, is going to be called 3-phosphoglycerate. Again, we have two of them. And now we're going to be changing it so that this phosphate is not on the third carbon, but we want it on the second carbon. And so the way we do that is with an enzyme called a mutase. So this is phosphoglycerate mutase. And so what that's going to do is it's going to take this and it's going to move it up here. It doesn't actually, you'll learn the mechanism for this as well, but it's not actually just directly moving it up there. It's actually trading phosphate groups with the binding pocket of that enzyme. But that you'll learn that in the slides when he covers it. So we've got 2-phosphoglycerate here instead of 3-phosphoglycerate. Two of them. And then we've got to get rid of, we're trying to get it closer to looking like pyruvate now. So the way that we do that, this enzyme is called enolase. And the important thing about enolase is that it's actually going to release water when it reacts. So we're getting rid of 2H2O here to make 2-phosphoenol pyruvate. And phosphoenol pyruvate, if you remember, is considered the quote-unquote king of phosphoryl group transfer potential. And that's because it can do this following reaction so readily. So pyruvate kinase, which is actually kind of named weirdly because we're not actually sticking a ATP onto anything. Or I'm sorry, sticking a phosphate onto anything. But what pyruvate kinase is going to do is it's going to take this phosphoryl group that's very ready to leave from phosphoenol pyruvate, and we're going to use that to make ATP from it. So pyruvate kinase will have 2 ADP, go to 2 ATP, and we're left with 2 pyruvates. And so this is actually the reaction that gets us the 2 net ATPs that we get from glycolysis, because you can see here that we invested 1 ATP there, we invested 1 ATP here. We get the 2 back that we put in right there, and then we get 2 extra. This is actually what makes it a process that produces energy. And so that's glycolysis. If you look at it like from a very, very zoomed out perspective, instead of just like focusing on all these tiny little reactions that I've just gone over, you can see that it's actually very intentionally made like this. Because if you look at these first reactions, we know that we're starting with a 6-carbon compound and we want to get it to 2 3-carbon compounds. So the way that I like to think about it is that these reactions up till here, these are like getting our glucose to look more like a symmetrical thing. We get progressively more symmetrical as we go down until we get this, which is like an actually symmetrical molecule, and then we can just split it in half. Once we split it in half, then we've got these 2 sort of different looking 3-carbon compounds that we can get to look like the same thing by doing the subsequent reactions here. And so that's how I like to think about glycolysis. A lot of people have asked me, in the past few days, just like how to study for this test and like how to study glycolysis and all this stuff. I think I don't want to say self-explanatory, but because it's really not self-explanatory. But if you write it out like 3 times a day, which is what I recommend you do, you'll not only like get it down really well memorized, but you'll also start seeing like patterns and things. And like that's how I was able to like tell you like, oh, the first half of this pathway is like, getting it down to a symmetrical molecule and then splitting it up into two of the same thing. You'll start seeing things like that, and you'll get like the hang of. You'll start developing your own mnemonics and things like if you have trouble in remembering an enzyme, just writing it out a bunch will help you remember. And so one thing that I did want to mention is because carbon tracking is super, super important for this. So this is the question from the Action Center that I wanted to go over. And so he's asking about, alcoholic fermentation, which releases CO2. And so the way that I want to think about this is that, so glycolysis, that's going to give me 2 of these pyruvate molecules, right? So pyruvate looks like this. So this part is going to go away as CO2. That's what gets released. Now, he's asking where these radio-labeled atoms would have to be located to ensure that CO2 is produced. So ensuring that CO2 produced means that both of these carbons have to be labeled, that we get from glycolysis. And so this is actually a little bit tricky when you start thinking about it. So I'll draw glucose first. Okay. So once we have this, what ends up happening to glucose, I'll copy it over here. Okay. So once we have our glucose, we're going to eventually have to like split it down the middle. And so we split it right here. And I'm going to switch to a different color for this actually. Okay. We split it down the middle. And now we have these two 3-carbon compounds. Now what ends up happening here is that this, when we split it, is actually going to be let me go back to red, CH2 OPO3 2 minus. This will have changed into our carboxyl, and then this will be, the carbons will change as a result, just because, we went from glucose to fructose 6-phosphate, fructose 1,6 bisphosphate. So this likewise will be CH2OPO3 2 minus. So this was glucose. This is fructose 1,6 bisphosphate. That's what the line structure of that looks like. And so when we split that, you can see that, so we care about these carbons right here, because this right here is the phosphate that belongs to dihydroxyacetone phosphate. This, the bottom one, is going to be glyceraldehyde 3-phosphate. And the easiest way to remember that is because glyceraldehyde 3-phosphate has the phosphate on the 3rd carbon, and this phosphate is already on the 3rd carbon, so we don't have to like mess with it and change it around. So the bottom will be glyceraldehyde 3-phosphate, the top will be dihydroxyacetone phosphate. Now, so as a result of that, we know that this will become, this carbon right here will become the aldehyde. And this carbon right here will become the aldehyde for glyceraldehyde 3-phosphate. And so if you count these carbons, so these carbons are going to be the ones, because they're the aldehydes, they will go through, as the aldehyde from here all the way down the pathway. And so you know that since these two are going to be your aldehydes, this is carbon 1, 2, 3, 4. So carbon 3 and 4, those are going to be the ones that if you label those, you'll have those, in the carbon dioxide produced.